I'm going to begin with just sort of an overview of why we should care about modifying plant genomes and some of the inherent challenges in modifying plant genomes with the various technologies that we've heard about, and particularly CRISPR-Cas. I'm going to give some examples of single gene modifications that we've made over the years, examples of NHEJ and HR-induced modifications that enhance trait or provide trait value to the crop that's being modified. And then, as we've heard throughout the week, delivery, as Dana always says, is so key. And so we've been working a lot on developing new vectors for reagent delivery, focusing on plant viruses. And I'll give some examples of NHEJ and HR-induced events with these vectors that we've developed. And very soon, you will all be consuming, very likely, products of genome engineering that are making it into the food supply. And so we'll talk a little bit about those issues, and as how they pertain to the sort of GM debate, the GM crop debate. And then finally, I'm going to tell a little story about how we've taken the CRISPR-Cas system and used it to confer virus resistance in plants, and then a little bit about future prospects, where we're heading. So the motivation for genome engineering in plant agriculture is really illustrated on this slide, which depicts the growth in world population that we're seeing. With 9.5 billion people by the year 2050, we have a lot more mouths to feed. As you can see from this graph, a lot of the population growth is in regions of the world, Asia and Africa, where the food supply is already tenuous. And it's important that we are able to produce the food to feed the growing population and also distribute it appropriately. Now, agricultural scientists have been very good about increasing productivity to meet the demands of food for the world's population. This slide shows increases in productivity for the major crop species, which have been, we've seen over the past several decades. But if you project onto this how we have to increase food production in the coming years, you can see that if these trend lines continue, we're going to fall short of available food in the very near future. And so, there are many ways, of course, that we're going to have to, many approaches we're going to have to employ to achieve the demand for world food. But it's clear that, one of them is that this rate of crop improvement must increase significantly. We also have to lower inputs. We have to develop plants that can grow on more marginal lands or with less water or less fertilizer. And so, in part, we're hoping that genome engineering can help, solve some of the solutions that are needed in the coming years. There's really two reasons why we engineer plant genomes. One is to understand plant gene function. It's just a basic biological question. There's a lot of genes in plants that we have no idea what they do in terms of the plant phenotype. And then once we do understand how they work, the idea is that we can harness genetic variation that's relevant to agriculture and produce new crop varieties that will meet the demand for food in the coming years. So, challenges in engineering plant genomes. One of them is this enormous amount of genetic redundancy. So here's the relative genome size of several of these important crop plants. So corn, the human genome's a little over 3 gigabytes, gigabases. So, corn sort of approximates what the human genome would look like. And even if you look at corn and soybean and rice, which have reasonably modest genomes, and they behave as diploids today, they've all had past episodes of polyploidization. So most of the genes are redundant, and there's remnants of these duplicate gene families. So when you target one gene, there's usually 3 or 4 or 5 others that can compensate for its function. So many times we have to target multiple genes. And then, so these species represent one spectrum, but then we have hexaploid wheat, which just has this enormous genome full of repetitive sequences, and the genes are, scattered throughout this repetitive, these very highly repetitive chromosomes, and they also present a targeting challenge, and breaking highly repetitive DNA can sometimes be a challenge for DNA repair and result in chromosome rearrangements. So redundancy and genome complexity are two of the challenges that we certainly face in plants. And the other is delivery methods. So most of the, when we heard talks of, engineering human cell lines, they're not unlike what's shown here on the right, protoplasts. We can take a leaf tissue and digest away the cell wall and release just these membrane-bound cells, and then treat them with reagents like polyethylene glycol, or we can electroporate them and deliver at high efficiency our genome engineering reagents into these populations. So these would behave like human cells in culture. And in some plant species, these individual cells are like stem cells. That is, we can get them to divide and grow, and we can treat them with hormones, and we can differentiate them into organs like leaves and then ultimately roots and entire plants. But it's only a very small number of species that we can do this with. So if you're a potato or a petunia or a tobacco plant, we can do this at very high efficiency. And as a consequence, we can make many types of complicated genome modifications to those species and get whole plants back. But for most of the rest of the plant kingdom, DNA delivery is a real challenge. We take partially differentiated organs, embryos, or leaf explants, and treat them with biolistics, blast the DNA into the cells, or treat them with agrobacterium, a soil bacterium that will deliver DNA to the cells. And only a very small subset of these cells take up DNA. And even smaller population actually get modified. And so we have to somehow identify those rare cells, coax them into growing, dividing, and differentiating so that we can recover plants with modifications. So it's a real challenge for many plant species to recover those that have targeted modifications. And it's time consuming. Going from a cell to a full plant can take upwards of a year. So yeah, DNA delivery is also a big challenge that we face. So I want to sort of just illustrate some of the types of modifications that I've made in plants and through this process sort of illustrate some of the challenges that I've just talked about and how they've been solved, and both delivery methods and how we overcome gene redundancy. I'm going to first just begin with non-homologous end joining, where we're introducing the nuclease into the cell, targeting a gene, and asking for a mutation to be introduced that will create a gene knockout. So as Dana alluded to, so I work at the University of Minnesota, where we sort of develop the technologies and try to optimize the technologies, but I also serve as a science officer for a company that is using talons in particular to develop new crop varieties. And so the example I'm going to give is a gene knockout that we created at Calyxt to create an agronomic trait of value. And our target species here was potato. So potato is one of those rare plants. We picked it because we could take a leaf, digest it, isolate protoplast, introduce our genome engineering reagents to single cells, and regenerate an entire plant back. So, we overcame one of the delivery challenges just by selectively picking potato. And we targeted a gene, an invertase, that's expressed in the potato tuber, but it's expressed when the tubers are chilled, so it's a cold-induced enzyme. And so if you buy potatoes at the grocery store, or you don't put them in your refrigerator, because when you do put them in the refrigerator, you induce the expression of this invertase, which starts to break down the sucrose into glucose and fructose. And as a consequence, you induce what's called cold-induced sweetening, so the potatoes begin to soften. And so that's fine for home use, but if you're an Idaho potato processor and fall harvest season comes around and you just have literally tons and tons of potatoes that you need to store because you can't process them all at the same time. And it would be great if you could, reduce the temperature to preserve the potato and inhibit the breakdown of sucrose to glucose and fructose. And secondarily, these reducing sugars, when heated at high temperature, which occurs when you make chips, for example, they'll react with free amino acids and create acrylamide. And so if we could knock out this enzyme, we could perhaps increase the storability of the potatoes, but also reduce the acrylamide in the fried potato products. And some of the states, like California, have set limits on the amount of acrylamide that can be present in fried foods. Many French fries and potato chips, particularly those delicious dark brown kettle chips. They're chock full of acrylamide, I'm afraid. And so California and soon the FDA will probably set standards for the amount of acrylamide that's allowed in fried and heat-treated foods. And so we also hope that our product would, have reduced acrylamide and fall below the standards that are likely to be set. So potato is a tetraploid, so we have 4 copies of every gene to worry about. And our invertase, of course, has 4 different alleles. So we made, in this case, talons again, although, CRISPR-Cas also works extremely efficiently in plants, and very similar to the many stories that we heard in animal systems. But in this case, we used talons, and we introduced the talon DNA by, PEG transformation into the population of cells. And we just started to get these cells to grow and divide into petri dish. They form what's called callus, this little clumps of undifferentiated tissue. And if you apply hormones, you can get them to regenerate first shoots and then roots. And so we invoked no selection. We got about 70 to 90% of the cells were transformed. We invoked no selection, and we just started screening plants that we derived for mutations. And so here we're looking at 102 regenerated plants, and 14 of them had 1 or more of the invertase alleles knocked out. 3 of them had all 3 or all 4 alleles knocked out. So we have homozygous mutants here in this invertase gene. And we were really excited when we got these first plants, and so we grew them in the growth room and harvested the potatoes right away. We saved 1 so we could propagate the plant, and we did fry tests on the other. And so these are examples of our knockout potato stored at 4 degrees for 2 weeks versus a wild type potato stored, same temperature, same treatment. This is a wild type stored at room temperature, chipped and fried at the same, in the same conditions. And you can see, the caramelization is indicative of those reducing sugars. And you can see when you actually do the sugar analysis that the knockout mutant, 4-copy knockout mutant, had much more sucrose and fructose and glucose, whereas the wild type, cold-treated wild type, the sucrose had broken down into these reducing sugars, giving rise to this caramelization. And then if you look at acrylamide levels in these chips, we saw about a 3.5-fold reduction of acrylamide in our cold-stored knockout line. So, we're really excited and optimistic then that through this simple gene knockout, we've introduced and created a trait that will have some value both for the producers and for the consumers in terms of improved storage and lowered acrylamide, respectively. So now we're sitting here with our potato, right? And we want, we just grew them in a pot in the greenhouse. We really, in order to see if a trait's going to have any value, you need to get it out into the field. You need to see how it behaves in the real world. And so, we'd like to start planting these potatoes outside. But if this is sort of a new way of creating and introducing genetic variation. So the biotech crops that have been produced thus far all have sort of foreign genes that have been added into their genome. And so the first step you have to do is to go to the regulatory agencies, particularly the USDA, and say, okay, I've created this transgenic potato, for example. Is it regulated or is it not regulated? And if it is regulated, you have to go through quite a bit and quite an expensive process in order to get it deregulated, costing tens to maybe hundreds of millions of dollars. And so we approached the USDA with our potato and said, well, we introduced a talon. We showed that there was the talon hadn't integrated into the genome, that it was just transiently expressed. And the only real change was that we knocked out that gene. We created a typical NHEJ small deletion at the cut site. And so after some consideration, the USDA said that the variety we made through talons is not regulated. And so that means we can immediately go out, and we have, and this year, in fact, we are planting these potatoes outside to see if, in fact, there's any other phenotypic consequences of our knockout. But it raises a question about, how are we going to regulate these new products that are made by this new technology? It seems if you just delete a gene, then the USDA considers it really not much different than if you created that mutation through EMS or gamma rays or X-rays.
What if the deleted gene has a secondary function of detoxification of something?
Of a secondary function of?
Secondary function to detoxify some endogenous chemical in the potato. So it's not important in the wild type because you never see the toxin.
Yeah.
But you delete the gene and you're thinking it does something to sucrose, right? But now you also have a build-up of the toxin, and without systemic analysis of metabolism, you're not going to know that.
Yeah. So the question is, what if there's other outcomes of the modification that we don't quite understand yet? So the USDA approval gives us the authority to grow it outside. In order to enter it into the food supply, we have to get approval from the FDA, basically the Food and Drug Administration. So they're going to ask questions like the one you just did. You know, is there a partial protein that might cause an allergic reaction in people who consume it, or is there some other buildup of some other chemical that you might not want in a potato that's consumed? So there's still other layers of regulation. And then the EPA, the Environmental Protection Agency, is yet another layer that would have to make a consideration. But at least in this first step, we can ask, is this potato even worth pursuing as a trait by getting it in the field and seeing how it produces and how the potatoes that it generates are stored, how well they store. Yeah. So, just recently, a few weeks ago, the Obama administration asked the USDA, FDA, and EPA to reevaluate regulation concerning biotech crops because, the old regulations were really sort of built around transgenesis, introduction of foreign DNA, and it's clearly now that we can make a wide variety of modifications to food crops, and we need reasoned and cautious regulation that also considers these different types of modifications that can be introduced. So for that example of NHEJ, we had single cells, we introduced the reagents, and then created the knockout mutation. As I said, that's only really useful for a handful of species. And so we try to think of ways to make it more easy to recover knockout mutations. And in one approach, we're harnessing RNA viruses as vectors. And so the idea here is, so RNA viruses, you can add small bits of additional information to an RNA virus. And as it infects the plant, it'll carry that information through the process of infection. So the idea is, can you get it to carry a nuclease or a guide RNA? And then as it's infecting the plant, it would then be mutagenizing all the cells that it contacts. And so then you could maybe either harvest a tissue and regenerate a plant, but more ideally and simply is if it gets into the germinal tissue, maybe you can just harvest seed, and that seed could bear the mutations, the NHEJ-induced mutations. So you don't have to do this cumbersome tissue culture steps. You can just infect, harvest seed, and obtain your mutant. So we tried this out initially with a virus that infects an RNA virus that infects a wide variety of plant species, tobacco rattle virus. And it has two RNAs, one of which encodes the polymerase, the other has a coat protein. And the second virus, or the second RNA, can be engineered to carry additional information. A limited amount. You can't get much more than, a kb and a half or so, depending on the virus. In this case, we could only introduce one ZFN monomer. We couldn't even include a dimer, which would need to be to cut a unique site. But in this test case, we had a plant that had this integrated reporter that had the monomeric site opposing each other, so that ZIF268, this well-characterized zinc finger nuclease, would bind to the identical sites, bring FOC1 into proximity, and create a break. And what we were scoring in this case were NHEJ-induced breaks at this reporter gene that was resident in the genome. And so this slide again just sort of illustrates. It turns out that the cleavage site is coincident with a restriction enzyme site, and so the wild type sequence would be cleaved, and that's what we see here. The cleavage products of a PCR product that encompasses the wild type sequence. But in tissue that's infected with the virus, if it's mutagenizing that site, you're going to destroy that cleavage site. And so you get this band that's restriction enzyme resistant, the PCR product that's restriction enzyme resistant. And then again, if you sequence that fragment, you see that it has mutations. So through this process of infection, we were in fact able to deliver the nuclease and to mutagenize tissue. And then more recently, in collaboration with Magdi Mafusa's lab at Coast, what we did is we engineered the virus to carry a guide RNA. We generated plants that were constitutively expressing Cas9, and so now as this virus was moving throughout the plant, it's delivering the guide RNA and mutagenizing the tissue. And in fact, we could achieve, at some, albeit low frequency, seeds that had inherited the mutation. It's still not efficient enough that you would sort of abandon the traditional approaches, but I think the results open the door for improvement. We can now think of using perhaps genetic backgrounds in plants that would allow more readily the virus to enter the germline so that we can increase the frequency of mutagenesis, sidestep these tissue cultured methods, and recover seeds just through cultivating the plants. So the next set of example I'd like to give is the process of creating mutations through homologous recombination. We're creating the break site and introducing variation that we're interested in. And this project is a project that's funded by the Gates Foundation to create herbicide-tolerant cassava. So cassava is a very important carbohydrate source for the people living in sub-Saharan Africa in particular. So it's a woody shrub. It takes about 1.5 years to mature, and it makes these large storage roots that are very high in carbohydrates, very high in starch. And so, as I said, this is the main carbohydrate source for many of the peoples of sub-Saharan Africa. Unfortunately, one of the big problems in cassava is the weed problem, and if you have 4 hand weedings per year, you can get yields that approximate treating the field with herbicide. But if you don't weed at a regular basis, you have a significant loss, 20 to 80% yield loss due to weeds. And then it's the women and children who carry out a lot of the work in the field. And so the humanitarian effort behind this project is to alleviate this work effort and ideally get the children into school and to be educated so that they don't have to work in the field. And so, almost 90% of the corn and soybean that's grown in the US is herbicide resistant. Almost all of the cotton is. So herbicide tolerance has been achieved, but it's been achieved traditionally through transgenesis. We've introduced a bacterial gene into the plant that encodes an enzyme. Actually, the plant has the very same enzyme, but the bacterial form is resistant to the chemical Roundup or glyphosate. And so traditional GM technology, we can now create these plants that are resistant. You spray the fields, and the weeds are killed, and your crop variety persists. But again, since the transgene that's added is also present in the native plant genome, our goal was to engineer the native plant genome to create herbicide tolerance and achieve the same ends, not through transgenesis, but through just a subtle modification of the plant genome. Now, we've done this before. In fact, one of the very first gene targeting papers we published targeted a gene that conferred herbicide resistance to a class of herbicides, sulfonylureas and imidazole, they're called, another related chemical class. And so this is the acetolactate synthase gene. It's involved in branched chain amino acid biosynthesis. So we don't have this enzyme. So we get all of our branched chain amino acids from our diet. And so it's a great target for herbicides because if you inhibit its activity, you don't harm animal life. But if you spray the upper Midwest with chlorsulfuron every year, you put enormous selective pressure on weeds. And so herbicide-resistant variants of this enzyme have emerged, like this W to L substitution. And so what we did is simply, at the time, engineer zinc finger nucleases to create breaks in the chromosome and introduce a donor molecule to create this amino acid substitution. And gene replacement frequencies, well, actually our best ZFN didn't cut very close to our intended, these would have been much better reagents, but they weren't very effective. Our best ZFN cut about 1,300 base pairs away. So we had to use a fairly large donor in order to convert this distant mutation. But still we could get, 2.5% of our transformed cells would incorporate the mutation. Now we're using TALENs or CRISPR-Cas. We can, of course, drop a very effective nuclease right on the target site and get higher frequencies. Well, the frequencies aren't so great here, but the donor is pretty small. But we can get much more efficient frequencies of recombination. And we can generate plants that are, in fact, resistant. So a single amino acid substitution, these are two plant varieties, the recombinant with the single amino acid substitution and the wild type grown on Petri dishes with the herbicide chlorsulfuron. And so what we're targeting now with this Gates project is the same mutation, but in the cassava genome, this W to L substitution. There have been a number of amino acid substitutions that also confer resistance. And then we're also targeting a gene involved in aromatic amino acid biosynthesis. This is the target of glyphosate or Roundup, the most commonly used herbicide. And here we need two simultaneous amino acid substitutions. And so, natural weed populations have emerged more slowly that are resistant to this chemical because of the need for these two amino acid substitutions to confer resistance. But our goal is basically to just swap out this exon and introduce the mutations to create the herbicide tolerance and ultimately, hopefully, improve productivity by better managing weed control and alleviating the work burden in cassava. But this also raises the question about regulation, right? In the potato instance, we just removed some nucleic acids. But now we're starting to do genuine editing. We're actually changing amino acid sequences. You could imagine that you would introduce variation that's already found in nature. For example, if there's an outbreak of disease, we often identify wild varieties of the species that are resistant and then cross the resistance genes into the plant. But if you know what those resistance genes are, you could just go ahead and edit the plant genome to confer that resistance directly. So there, the end product is very similar to as if you just initiated a breeding program. But what if you're going to create an enzyme that has a novel activity that's not found in nature? How are you going to regulate it? It's going to go into the food supply. Are there other consequences for that enzyme being expressed in the plant? Those are all questions that have yet need to be answered. So where we use viruses to sort of make it more easy to achieve end joining, that was the strategy where we infected and harvested mutant seed. We're also using viruses to enhance gene targeting. And in this case, we're using a class of DNA viruses, not RNA viruses. These are called the Gemini viruses. And they have circular DNA genomes. And again, because they have a DNA genome, they can encode the nuclease, and they can also provide the donor molecule, the repair template, to introduce a mutation in the genome. And when these viruses infect plants, the infection is transient. They amplify to very high copy numbers, and then they go away. So they can give you a burst of genome engineering. You can imagine them giving a burst of genome engineering reagents during infection, allowing you to achieve your modification. So what we did in terms of engineering these viruses for genome modification is we modified them. We rendered them no longer infectious. So this is a schema of the Gemini viral genome. It encodes a replicase, a movement protein, a coat protein, and it has these long intergenic and short intergenic regions, which are important. They drive expression of these proteins and are important in replication. And so we took the virus and we deactivated it. We changed it from an infectious agent into basically just a DNA replicon. So we swapped out the movement protein and the coat protein and put in our nuclease of choice and our donor molecule. So this could be a zinc finger nuclease, a TALEN, or CRISPR-Cas and a guide RNA, for example. And so the idea is that we would deliver these reagents to plant cells. And You could do this through biolistics. What's shown here is the use of agrobacterium. So agrobacterium will have this replicon sitting encoded on the TI plasmid. It'll get excised when it comes in contact with the plant cell and delivered to the plant cell. And then the tDNA is linear, but if we provide the replicase protein, it'll circularize and make that replicon. And then it undergoes rolling circle replication and it amplifies, often to excess of 1,000 copies per plant cell. So now you have an abundant source of the nuclease. And again, you can code any type of nuclease you wish, which will find the site in the genome to modify, create the break. And then the replicon also provides an abundant source of the donor. So you can then repair the break and incorporate the modification into the genome. So we tested this again in a simple test system with an integrated reporter gene in tobacco, one of our favorite models. So this is the reporter that's integrated into the tobacco genome. It has a very strong promoter driving coding sequences for beta-glucuronidase and neomycin phosphotransferase. So this is an enzyme that allows you to stain tissue histochemically. So if beta-glucuronidase is expressed, your tissue in the appropriate substrates will turn blue. And of course, neomycin phosphotransferase will give you resistance to agents like kanamycin. So this was designed to be a fusion protein, but we removed 600 base pairs and replaced it with a zinc finger nuclease target site. And so the idea is that the replicon would encode the nuclease that would break the reporter, the integrated reporter here, and there's also a donor with 1 kb homology arms and the missing 600 base pairs. So after recombination, you should reconstitute this reporter, giving you both beta-glucuronidase activity and neomycin phosphotransferase activity. So the way we did this, with the way we delivered these reagents, is into leaves. We just infiltrated into the leaves that agrobacterium strain. So it just moves in the leaves throughout the spaces between the leaf cells, and it infects all of this, or many of the cells within that leaf tissue. And then we can wait several days, and then we stain it for beta-glucuronidase activity. So these are some controls. So these are little sections of the leaf that have been stained in beta-glucuronidase activity. This is just what agrobacterium normally would deliver, the tDNA. It has the nuclease and the donor, and occasionally you get these little dark spots. These are cells in which we've repaired that integrated reporter, and they're now staining positive for beta-glucuronidase. So this is our replicon. So it has the cis-acting sequences that should allow it to circularize and replicate, but it doesn't have any replicase. So in effect, it's pretty much the same as the tDNA. And again, you see a few occasional spots. But if you add the replicase protein, you get the circularization, the amplification, and then when you stain that leaf tissue, you see an abundance of these cells. So you increase very significantly the frequency of homologous recombination in those cells. And we can image these tissues and quantify the enhancement in recombination. So these are 4 different lines of tobacco plants that have that reporter integrated at different sites. And we're comparing the standard method of DNA delivery, agrobacterium tDNA, and then we're using agrobacterium then to deliver the replicon and the replicase protein. So you can see in all of these cases, we get a great enhancement, 25, in some cases, excess of 100-fold increases in frequencies of homologous recombination. We spent some time to sort of tease apart, what's the most important element to see this in enhancement. It appears not to be the nuclease. The nuclease seems to be in good abundance. That is, you don't need the nuclease on the replicon, and you'll still see almost comparable levels of enhancement. It seems the 2 contributing factors is, one, the abundance of the donor. So the replicon, by making a lot more donor available, achieves half of the increase. And then, interestingly enough, that replicase protein that allows that virus to replicate and amplify, it actually has a pleiotropic, or has a secondary function that impacts the plant cell. That is, it pushes the plant cell into S phase. And so by going into S phase, you naturally induce the protein's enzymatic machinery that carries out homologous recombination. So you provide an abundant source of donor, and you also sort of change the state of the cell so that now homologous recombination is more conducive. And those two processes are what really lead to that big enhancement in recombination that we see. So, okay, tobacco is a great model, but we went to its cousin, the tomato, to see if we could apply this in sort of a real world setting. And tomato is nice. It's fairly easy to manipulate. It's fairly easy to deliver DNA to, and the genome has been sequenced. We know a lot about it, and it has some relevance in terms of as a crop species. So, but again, we wanted a simple readout to see if our Gemini viral replicons were modifying the tomato genome appropriately. And we chose a transcription factor called ANT1. And ANT1 regulates the expression of many genes that are involved in anthocyanin biosynthesis. So the anthocyanins are the purple pigments. And these are transgenic tomato plants that overexpress ANT1. So they have basically purple pigments in all organs of the plant, leaves, flowers, and fruit. And so we thought this was a great readout, because if we could elevate ANT1 expression, we could identify the cells that have undergone recombination. And our strategy was pretty straightforward. We just simply designed a CRISPR or a TALEN that would recognize a site in the ANT1 promoter to create a targeted break, and then a donor molecule that would knock in a strong promoter to drive ectopic ANT1 expression. And we also put in a resistance cassette so that we could enrich for cells by growing on kanamycin NPT2. And so, again, we delivered our replicons to the plant cells. In this case, we couldn't do it to individual cells, or we couldn't infiltrate it into the leaf, but we took, just after tomatoes germinate, we took the first leaves, the seed leaves, the cotyledons, and infected them with agrobacterium and put them on tissue culture. And these are the number of explants, the number of leaf tissues that we looked at in this particular experiment. So without a TALEN, we didn't get any purple tissue. If we didn't have replicase, we didn't get any purple tissue. But about 5% of those tissues treated with agrobacterium that delivered the replicon, we could see this purple tissue emerge, and then we could excise it and regenerate purple plants that had the modification. So again, it gave us a nice enhancement. The replicon allowed us to achieve our HR event, our gene targeting event. So Dirk talked a little bit about the importance of looking at the fidelity of recombination, and that's in stem cells, and that's certainly true in plants as well. So we generated a number of recombinant plants that were, that had the phenotype we wanted, and then we started surveying them to see what the integrity of the integrated locus was. It at ANT1 as expected, and did it have any unanticipated rearrangements or additional incorporation of additional DNA. So this is just a PCR survey. The plants didn't have any of the replicon anymore, so no negative amplification for the replicon. So again, I mentioned it's a transient. It replicates for a period of several weeks and then goes away. We did a PCR control. Primers 5 and 4 amplify the ANT1, so the tomato plant has this locus, this native locus, and so we get a positive PCR. If we do the right junction, primers 3 and 4, so one of the primers is specific to the promoter we introduced, and in all the plants we regenerated, we got this junction, but in a handful of plants, when we looked at the left junction with primers 1 and 2, some of the PCR reactions failed. And if we did a southern blot on some of this tissue, what we found is that here's the native ANT1 locus, here's a gene targeting event, so it's not present in the wild type, and those that failed, the left junction PCR had additional DNA. And then this is an example of one of those events, one of these sort of one-sided gene targeting events. When we looked at it, it had just incorporated part of the vector, the TALEN, part of the TALEN coding sequences in additional DNA. And so upwards of a third of the events that we recover through HR have these types of events, not unlike what Dirk described in human stem cells, imprecise repair. And so you have to be careful, you might get the phenotype you want, but be careful that there's not any additional DNA that's been incorporated. And then those purple plants that we made, we could then, monitor transmission into the next generation. So these are seeds. Interestingly, we could only do one allele. We could never achieve a biallelic modification. And it was in the next generation that we realized why. They would segregate wild type and mutant, and if they segregated a homozygous, gene targeting event, both alleles had been targeted, the plants were really stunted and never really developed. So we compromise plant health by overexpressing these anthocyanins. So it was a partially deleterious modification. A story I want to tell now is a little bit different. This is CRISPR's 101, right? The CRISPRs are an immune system for bacteria and archaea that prevent, DNA invaders from, infecting the bacterial genome by clipping up and removing the DNA. So we reason that, we've been thinking about the Gemini viruses, DNA viruses that infect plant cells, and we harness those viruses to deliver our genome engineering reagents, but those viruses are actually really important plant pathogens, causing billions of dollars in crop loss every year. And so the notion was that, maybe we can just confer this immune system, this system that confers immunity against viruses, maybe we can just introduce it into the plant genome and confer resistance against these DNA viruses, these Gemini viruses. So this is that bean yellow dwarf virus that I already described to you. Again, the long and short intergenic regions, coding sequence for rep, code protein, and movement protein. What we did is we engineered a number of guide RNAs that recognize highly conserved sequences within the virus genome. There's a lot of variation among these viruses, and so we wanted to target regions that were going to be conserved amongst most of them. Some of them are cis-acting sequences around the site of initiation of replication. Others are highly conserved domains within the rep protein that are important for rep protein function. And we made then a plant, in this case a tobacco plant, that was constitutively expressing Cas9. And in our initial tests, we took code protein and movement protein and again replaced them, in this case with the EGFP expression cassette. And our initial strategy was just to take 2 strains of agrobacterium, one which would deliver, in this case it was a wild type plant, so we were delivering Cas9 and our guide RNAs on one tDNA, and then in another tDNA we were introducing our replicon that had GFP in it. And so we infiltrate both strains of agrobacterium into the leaf, and then we image GFP. And so here's the GFP intensity you see if you just introduce, the agrobacterium strain with, or these 2 strains, but the guide RNA doesn't target the virus. And so you get, you normalize GFP intensity to that control. And then these are the intensities observed with a number of different guide RNAs targeting that viral genome. And in this case, the lowest readout was when we had 2 guide RNAs targeting 2 different sites. So this was making us feel pretty good that we could knock down replicons. And we did another experiment where we replaced GFP with an ampicillin resistance gene and a coli-1 replicon. So we took the regions that were infiltrated and just made DNA and then just transformed E. coli. And you see that the number of circular molecules that could be recovered in E. coli correlated very well. The colony-forming units correlated very well with the fluorescence. They were basically the same. So the knockdown in fluorescence was likely due to the reduction in GFP expression due to the reduction in these circular replicons that were formed. You could argue here that what you're really measuring is Cas9 cleaving the tDNA, and the reduction is because you're just killing a lot of tDNA before it actually circularizes. So we wanted to do an experiment to show that we're knocking down viruses themselves. So we took, in this case, a Cas9 constitutive plant and infiltrated our viral construct here. And then we waited 3 days and then infiltrated a second region of the leaf with a couple guide RNAs. So the idea is there's been enough time now for this virus to start to move throughout the plant, and where the guide RNAs are delivered, Cas9's everywhere, the guide RNA should be chewing up the virus and not the vector we're using to deliver the replicon. So we can sample control tissue and test tissue and then look, in fact, whether or not we're actually cleaving the virus. And so these are results from a couple different plants. These are PCR amplicons. These are 2 guide RNAs that cut a little over 300 base pairs apart. So this is a PCR amplicon from an unmodified site. But in this tissue where the guide RNAs have been delivered, we're actually seeing these deletion products, meaning the virus has gone from here to here, and we're actually cleaving the virus, and again, not just the DNA that we're delivering to these cells to generate the virus. So that was encouraging. And then, of course, the real readout was when we looked actually for virus throughout the plant, and we could see that if we look at viral load by qPCR, we can see in Cas9 only versus those that are expressing Cas9 in the guide RNA that we have a significant reduction in viral load. And then importantly, the plant phenotypes are reflective of improved resistance to these viruses. So these are tobacco plants. The lower leaves were infected with agrobacterium, infiltrated, and that launched the viral infection that then over the course of 21, 28, and 35 days had a chance to move throughout the plant. You can see by 35 days we're getting these yellow crinkly leaves, which are indicative of virus infection. But in plants that are expressing Cas9 and a guide RNA targeting the virus, we see attenuated phenotypes. So more consistent with the reduction of viral infection.
If you go back 2 slides, you had 3 plants there. Was there any difference between those, or is just kind of just natural variation in our new system?
These are just different transgenic lines expressing Cas9 and the guide RNAs. So they are independent events. So what we should do is go back and see if Cas9 is being expressed at the same levels in each of those independent events, because they're landing at different sites in the genome. You could imagine some just position effect variation in terms of expression. So I think that's the variation that you're seeing there. So we've now made tomato plants that express Cas9 and some guide RNAs targeting a tomato Gemini virus, a virus that has a pretty big impact on tomato production. And we have a collaborator who the virus is normally transmitted by flies, white flies that feed on the tomato plant. So rather than forcing the virus in through agrobacterium, we can just ask with these tomato plants and the natural vector for the virus whether or not we can confer resistance in greenhouse-grown tomato plants. So we're optimistic that we can borrow this innate immune system from bacteria and help alleviate infection by these DNA viruses. Okay, so I'm just going to, in the last few minutes, just talk a little bit about some future prospects for genome engineering in agriculture, questions that we think about and are beginning to tackle. Everything I talked about has been one gene modification, a single knockout or a single gene replacement. But I think the future is all going to be about multiplexing, changing many genes simultaneously to introduce genetic variation. So, I talked about, the need to increase the world food supply. Well, there's a lot of studies that people have carried out where they've been trying to identify traits that confer, in this case, salt tolerance to rice. So you can grow rice in soils that are normally not conducive to rice production. But these complex phenotypes, drought tolerance, disease resistance, many genes act together in concert to confer these complex traits. And so quantitative trait mapping has been carried out in a number of species to identify regions of the genome that have genes that work together to confer these complex traits. And so what we're thinking about increasingly is QTL engineering, right? These are still oftentimes megabases in length. If you have really rich mapping populations, you might narrow it down to 10 or 50 genes that you could think about knocking out to identify the one that's actually conferring the variation of interest. But if we can take the two parents, for example, that are on the extremes of the phenotype and think about moving a whole domain of a chromosome from a variation from one parent into the other to basically homozygous, a large region of the chromosome, we might be able to think of strategies, genome engineering strategies, that would allow us to narrow in these large regions of the genome wherein we know genetic information of relevance lies, but we just don't, we don't have the resolution to focus in on it. So I think there's a lot of opportunity for genome engineering to help us identify genes of relevance for agriculture. Sort of along these lines, if you think about the domestication process, so for 10,000 years we've been selecting plants that have traits that provide more food or are more resistant or easy to grow in the field. But now that we have so many powerful genetic approaches, we can sort of ask how many genes are actually really important in the domestication process. So teosinte, the wild progenitor of modern maize, there's really only 5 or 6 genes that are the major contributors to this huge phenotypic change. And so we can now think about just testing that hypothesis, going into teosinte, for example, modifying those 6 genes and seeing what the heck does that plant look like after we make those modifications. Are they really the major contributors to domestication? And there are many species where only a handful of genes seem to be important in domestication. And then you can think about, well, maybe you can then go to other grasses, for example, introduce those same changes and confer traits to plants, for example, that might grow on more marginal lands and that are not currently, that could maybe be made more productive for animal food or fiber or whatever the desired commodity is. So molecular domestication is another thing that we think about quite a bit, not unrelated to the QTL engineering sorts of approaches. And then finally, we can start to think about making plants that do things that we never thought possible before. And actually, I have to give credit to the Bill and Melinda Gates Foundation. They have the resources and the vision to really start thinking of taking on really challenging projects. So legumes, beans, peas, alfalfa, they have natural symbioses with soil bacterium, and they can take up wheat, or they can take up nitrogen from the atmosphere, and in collaboration with the soil microbes, they can fix that nitrogen and incorporate that nitrogen into the plant material. Of course, most plants, like cereals, we have to fertilize in order to provide the nitrogen. But there are projects being funded now to think about just engineering, for example, wheat, to fix atmospheric nitrogen to overcome the need to provide nitrogen, which derives mostly from fossil fuels, and is a big problem of pollution in terms of runoffs into streams and lakes. But you can think about it. You have to engineer a symbiosis with a bacteria that doesn't exist. It happens, yes, in some branches of the plant kingdom, but that's a big leap of faith that it can be accomplished. And clearly, genome engineering is going to be needed to achieve those ends. And another big project is the C4 project. So, the enzymes that fix carbon dioxide from the atmosphere and turn it into carbohydrates are pretty inefficient and are inhibited by atmospheric oxygen levels. And so some plants become much more efficient, like corn, for example, because they sequester carbon dioxide into differentiated tissues that have much less oxygen, so they can much more effectively fix atmospheric carbon dioxide. And so can you turn rice, which is a C3 plant, into a C4 plant? You may get 20, 30 or more % increases in biomass by just making photosynthesis more efficient. But again, you have to change the anatomy of the leaf, engineer a wide variety of the expression of enzymes in order to achieve these very complicated phenotypes. But they're now can be imagined with genome engineering and the various tools of genomics that are now available. So I'm just going to close there. Much of what I talked about was work that was done in my lab at the University of Minnesota, with the exception of the potato project, which I mentioned was done at Calyxt under direction of a former postdoc of mine. So thanks. I'll be happy to answer any questions.